Good afternoon, everybody. It is Wednesday and the 3rd of April. You're here at Lunch and Learn. Today we have our special guest speaker, Dr. Lew Lim. He has been with us before. He is the CEO and founder of Vielight. A lot of you folks work with Vielight. Today he's going to give you an update on some of the photobiomodulation techniques they're using with brain functions with an emphasis on Alzheimer's. You've read his background. He's been with us before, so I'm not going to give you any special introduction other than Lew. Thank you so much for being with us, and it's all yours.
Okay. Actually, Rob gave me a couple of questions. One is to explain all these, new Vielight devices, and I will put in the context of photobiomodulation rather than, well, I'll try to do both, but also how photobiomodulation can address dementia. So really, it is really in two parts. Now, I just want to start with the devices. I think I will show what they look like, because some of you may not have seen the real products. And I will take some questions maybe in this section, and then in case you forget what they are, and then go on to the dementia part, where I will also take another set of questions. So those who have been with us, following us, will be familiar with the various stages. Maybe those who have got the earlier versions, sorry about that. We keep refreshing and innovating and updating our products. So at the moment for the neuro, anyway, we're at version 4, which is on the right hand side. Now we started with the neuro 1 a number of years back. Then we changed it somewhat because we changed the module from something pretty clunky to something smaller. We improved actually the management of the module. Then we did Neuro 3, where we shifted the controller from separately to the head. And we miniaturized that. And then some people complain it's not that comfortable because of this, these stabilizers on the sides. Then we are now in Neuro 4, where we added one extra module, and it's generally the context better, and the actually my updated picture had a close-up of this module, but generally speaking, all the LEDs that we do are not off the shelf. We actually engineer the LEDs so that we get the power that we want. We can go up to if we get the LEDs off the shelf, slap it on the back of a helmet or something, it's about 22 milliwatts, and then that is on contact. But a number of actually most devices out there cannot get a contact. So when you actually we measure everything. When you actually measure it, your off-the-shelf LED is going to give you like 5 milliwatts. And if you go into the daylight, you get a lot more power. But we can deliver with the Neuro 4 up to 300 milliwatts if you want to. There's multiple times. But we control it for the time being up to 100 for safety reasons. So for Neuro 4, we added one to the cerebellum. You can move it up and down to the visual cortex. And the idea is, I personally think the cerebellum is important for traumatic brain injury. All the latest research are indicating that, and the Alzheimer's and so on. So we used to have an extra called X Plus to edit it. We just removed that and just integrated everything to one without increasing the price. So that's a quick rundown of Neuro 4. We have now a version in black because a lot of consumers want something really, sexy, especially the athletes we're dealing with. So we have different versions. Now we want to see what it looks like. Well, I can't see myself on camera, but I hope you can see this. This is the Neuro 4. But we have now, actually, the logo is going to be in red. Okay, I have a few products to go through, so I won't spend too much time on this. Now, I wanted to show you this because if any one of you follow UFC, to some measure will know this, that Ilia Topuria just won the world featherweight title with a record of 15-0-0. He beat the previous world champion who held it for like 2 years. So he's been using these quite religiously, and he's been appearing on Instagram and so on. But he was using the version 3. Now he's got a new version 4. Version 4 looks like that. That is on this scary guy. Now we also released some. I know some people calling because of the customer service telling me about the vagus stimulation device. This is the sample. It is the first vagus stimulation device. It's been tested by a few people in ISNR. Nancy Victon's husband is one of them. And I say generally we have good reports. We haven't quite managed to do a proper clinical study because of whatever reasons. But it's out there as a general wellness device too. So let me see, I have it here. If you haven't seen this, this is the Vegas. Put it here. It powers 100 hertz, one to the nose. So that is an update on that. Now we came up with MIP. So the intranasal was original. You know, I hold the patent for that, and it's been helping a lot of people in many ways. Now you can use them both. So you can see it's connected to, we have 2 now, and we can have 2 applicators at the same time. And I have here I have the 633 red and the blue. Blue is antimicrobial. I have a lot of good reports on helping with, you know, actually even people with acute COVID and so on, and with people having cold recovering more quickly. This is one that many of you are familiar with, the original 633 for systemic effect. Then we have the others, the 810 for the brain, which can pulse. There is a intelligence in there, in the little thing like this, which can sense that when you have an 810, you can pulse it at 10 hertz or 40 hertz by just keeping a finger on the button. So that is supposed to go to the brain, and instead of having the big neuro, you can have a little device like this. Of course, the power is a lot less. And the 655, which is a laser, does similar to the 633 systemic effects. So Ilya Topuria, actually, we didn't know he was using this. He's been quietly using this, and he came up to us later talking about it. So he's been training with this for quite a while before he started getting onto the Neuro 3. Right now we then have this device that we used in our COVID-19 acute COVID-19 study. We have a long COVID study just in the site. We are analyzing the data right now. That has been approved by Health Canada. Now we have been given the approval for not just COVID-19, for upper respiratory symptoms, which means that like HSV and flu and a bunch of other things, COPD. So then we have a pain device that we haven't actually released yet because this is a medical device registered with the FDA for treating focal pain. Let me see if I have it here. So I still have the prototype. So basically, runs on a power bank. There's a strap for you to wrap around the joints and so on, like this, the power bank here. It's portable. It is actually, for a number of years we showed the original prototype pre-COVID, and people had good results and was chasing us, but we really wanted to get it right. We wanted to get all the approval because this is a medical device. We are just waiting for some formalities, actually an audit before we release it, anticipated to be in summer of this year. And then our flagship device, the Neuro Pro. This is the version 2. The version 1, I think some of you have. The version 2 is needless to say, we try to improve the. I'm not going to put this on my head. But this is the version that even I don't have it because the wait list is so long. We've given out everything right now. And if you want to learn more, there is a community. I think you can go to the NeuroPro web pages, get a link, and show your interest. You can join the waitlist. We anticipate now we're just finishing. There's some software in there. Actually, is mainly for testing. There will be some pre-programmed protocol to help you with understanding it and helping with the brain condition and stuff like that. You use a cell phone that's given to you, that's paired with it, and do the various customization, inputting your own protocol if you like. So it just takes a little bit of learning, but I think it's all worth it because you have a chance to do whatever you want. I think some of you are probably on the wait list. But anyway, I will not talk too much because this can take up a lot of time to talk about NeuroPro, but it is there. You can learn a lot more and contact our team. So yeah, I just hold on for the time being and go backtrack. Let me take some questions before I move on to the next stage. Anybody? Does anybody have any question on the devices?
We tend to have to unmute people, but if there's no one responding within the next minute or so, then I think they're good for now and want you to carry on.
I'll continue and then we can open up the Q&A towards the end. Is that okay? All right.
Perfect.
Now, I just published this paper about a month ago, and I don't know if any of you have a chance to read it. It delves deep into the pathophysiology as regards to traumatic brain injury. Why am I talking about this when I'm supposed to be talking about dementia? It is some of the, actually I've just submitted another paper, something similar, looking into the pathophysiology of Alzheimer's disease and how, breaking all this in the various aspects of it is tackled by photobiomodulation in various studies. Let me show you an example. In traumatic brain injury, you can actually break down to the various aspects like mitochondrial dysfunction, the reactive oxygen species, where it creates oxidative stress, axonal degeneration. This is, common with Alzheimer's disease, obviously. The autophagy, where your cells are supposed to die and get refreshed, and the lysosome is supposed to release peptides, the organelle in your cells, to clear this debris and unwanted byproducts in your brain. Then it helps with controlling the apoptotic cell death, which is programmed cell death in your cells. Then neuroinflammation, excitotoxicity, axonal injury. What is really interesting is when I talked about traumatic brain injury, there's no recommended or no FDA recommended medication to treat that. Is multifaceted, is complicated. But the thing interesting about photobiomodulation is it is one intervention, but it comes out with addressing something like this, an Alzheimer's disease, from multiple angles and tackling this physiologically aspect independently. So have a read because it's published. I think the next one will be hopefully be coming out soon. Now, Alzheimer's disease is also multifaceted, and the way I look at it, this is actually submitted in the paper. But look at photobiomodulation, how is relevant first. But you know, the big thing is really in the pathophysiological model, tackling it from various angles. And at the end of the process is the reduction of Alzheimer's disease symptoms. So this is what it looks like. It's complicated. Alzheimer's disease is a lot more complicated than people think. When FDA approved a drug like Leqembi, it's targeting actually only on, well, the main outcome is, the main endpoint is really the amyloid beta marker. So, and from that, okay, one thing about FDA is they want to see, nowadays anyway, as compared to a number of years ago when we first approached them, they want to see an objective measure. So these are really quite a big burdensome marker actually, because you have to go do a PET scan and stuff like that. But the thing is, that drug now, it's supposed to slow down the decline by 27% over 18 months. But that's only looking at one marker. But Alzheimer's is complicated when you look at the amyloid beta, which is like the middle on the left side. It really, you know, you're really missing on quite a number of things. And a lot of it starts from the origin, the genesis. I call it the pathogenetic factors. It can be from outside, like infection, environmental, they contribute to it. They can be genetic, which is, to me, actually, this is, I call it the endogenous factors. This is probably the biggest factor influencing Alzheimer's disease, this genetic predisposition. Then you have the other dysfunctions in blood-brain barrier, gut-brain, your gut-brain microbiota, your microbiome, the lymphatic flow, the metabolic syndromes, like, you know, what's the effect on the lipids and stuff like that. Then it shows in the biomarkers, amyloid beta is one of them, the neurofibrillary tangles, NFTs, the tau, originating from tau misfolding is another one. There's energy deficit, oxidative stress, calcium overload, inflammation. There is a group that says inflammation is really the main thing. You know, everything comes to that. But these are markers, and it leads to the precursors of Alzheimer's disease, which is the loss of neurons, synaptic connectivity, and the loss of volume. And then you lose memory, your cognition, your behavior. Now, what's really relevant in our field is it gets presented in the changes in brain networks. It gets presented in the changes in your, you know, the frequency, the brain oscillations and frequency. But I put a star on each one of these because there are studies out there, whether it's done by us or somebody else over the decades, showing that photobiomodulation, by directing the right power density in the right areas, pulsation, if you like, these are the stuff that I will show a little bit more. They have an influence on all these various facets collectively. That's why I believe, that eventually when we do our work, and we have to do more research, come up with the right set of parameters, and I'll touch on this later on, in personalization. I think it's potentially will have a big impact on the disease. I did a systematic review on what's out there. There are not too many human studies that's been published. There are, you can count them literally, 2, 4, is it 7? And I say it's heterogeneous, the parameters are all over the place. But even the sample size from one case to that one isn't with it, 37, 3 dropped out. And I say I question some of the quality of some of these studies, but I say generally, they're all peer reviewed. But I say that generally for sure, they're all safe, right? And generally, they include the improvements in cognition and memory. You got other outcomes like sleep and so on. And the most recent one, beginning of this year, I think is done in Iran. Didn't see that much in some of these measures, but saw disability improvement. So it calls for more studies. Yeah, so I'm looking at what it can inform us to improve in parameters that we do and so on. So what have we done? We actually do a lot more than just clinical studies. We want to see what are the effects on your cerebral blood flow and the blood perfusion and so on. So this is a study that's ongoing at Baycrest Hospital. They are affiliated to the University of Toronto. That's the machine we used, and that's our engineer subjected to the experience. With one laser on the forehead, one in the nose. But looking at just transcranially, we put one on the right. I got quite a lot of images. This is just the preliminary. We are doing 42 subjects. I think we're kind of halfway through right now, but in the preliminary data, we found that really there's a window for power density. It cannot be too little, it shows nothing. Too much actually shows negative. And say, for this, we are looking at, the window of 150 milliwatts for this particular person to get that level of response in the brain. And you won't find this level of response. I'll look at the work done in, say, other form of brain stimulation like transcranial magnetic stimulation and electrical stimulation. I have not seen anything to this level. So this is to the right, prefrontal cortex. And now we add the left, we see a lot more. This is real-time imaging, minute by minute. So here's minute 3, minute 4, minute 5, minute 6. So within minute 3, this group is at Baycrest, they're pretty good technically. Not too many radiology groups are able to see, within like 3 minutes. They usually take a bit longer, but you can see this response in the brain within 3 minutes, 4, 5, and 6. So that is promise of what is coming and we want to finish hopefully by the summer of this year, 42 subjects. How about EEG, our territory? Again, this is ongoing. We delivered gamma in the neuro. Here's the baseline. A, B is 5 minutes before treatment, and here we deliver 40 hertz at a level C, and then looked at 5 minutes after we stop, and then 15 minutes. We have a few subjects done, and it is not too far from what we are noticing here. So this person had, 5 minutes before somehow his gamma dropped. But when we did the 40 hertz stimulation in real time, we see the big surge in the gamma. Then we stopped. 5 minutes later, everything kind of synced back. But very interestingly, 10, 15 minutes after we stopped, actually the brain improved again. But the gamma, after we delivered, remember we delivered gamma, and 15 minutes after we stopped, the gamma was still showing strongly in the brain. So what does it mean? If you, so we've done gamma so far, we're doing the other frequencies. We have done it, and we want to, actually we are repeating some of the studies we've done earlier to make sure that we get more robust data. But I can conclude this so far from the preliminary findings is that when we deliver gamma, you'll get gamma in your brain. Whether you are looking for gamma because you want gamma in the brain to have higher functioning, or you want gamma in the brain for to treat Alzheimer's disease and so on. So that's ongoing. Now, the next generation of treatment is going to look at, for us, now you can treat Alzheimer's with a device in many ways, but what is kind of easy for a device that can be treated at home, I think, is to approach it from the networks and the waveforms. Now, in Alzheimer's disease, gamma seems to be really in the discussion right now, and gamma is supposed to have reduced, beta amyloid markers. The very latest finding is that gamma actually help with lymphatic flow because they found the actual cause is because it stimulates a certain neuropeptide called a VIP that opens up this astrocyte in the endothelial cells to well, that's endothelial for blood circulation, but also the aquaporin 4 opening the astrocytes to improve in lymphatic flow. Now we can look at many aspects. I have separated, and this is going to come out in another paper in the future. Some things are inelastic. You got to decide whether you want to use laser or LED. You got to more or less decide whether you want to use 808, 810, or 1064. That wavelength or that wavelength. Positioning on a head, that's relatively inelastic when you try to make it a home use device. Then the power density is elastic. You can change the power density. You can change the duration. You can change the pulse frequency. You can change. This is new. Some of you are into cross-frequency coupling. We're looking into embedding, say, 40 hertz into 4 hertz. Very interesting. You can look at the network, default mode network, and personalization. Now, very quickly, someone presents to you on the mechanisms. Very often, something like this comes out: effect on the cytochrome C oxidase in the mitochondria, effect on the electron transport chain. Now, when we talk about whether we should use 810 or 1070, pay a little bit closer to this, because according to this group, which is a very respected group in photobiomodulation, and I think it's about time it needs to be updated. This was published, I think, in 2016. I think it doesn't show more what we discovered. So questions are 810 or 1070. Look, 810 in this literature affects the mitochondria and therefore the release of nitric oxide and go on to the activation of the transcription factor from there. Here we are looking at the influence of the cytochrome C oxidase, which is in the mitochondria. When we talk about 1070, according to this group, you are really into this mechanism and not this other one. And here you are looking into the activating the heat and light-gated ion channel, the RPV1 or the heat shock protein, and therefore you are then creating these mechanisms to in the process of treating the brain. So now next question is that is a decision that we are doing more investigation. We may incorporate both, but we really want to understand what is the correct thing to do. So just watch out for that. Now the question is 40 hertz the best? The literature is really strong on 40 hertz, but I'll say hang on a minute. Is the power of the of institutions like MIT. There's such they did really good work. I mean the quality of the literature is really very good. They're published in Nature, but everybody seems to follow. Okay, 40 hertz, 40 hertz, 40 hertz. But there's a group in New York, a very respected group headed by Buzaki. You say, hold on a minute, I'm replicating the work done at MIT, and the 40 hertz is, well, I'll interpret does not work means it does not seem to produce the endogenous gamma in the brain as you claim to be. And when we measure the change in the amyloid beta, it's not the same. It doesn't have any significant change. And then there was a rebuttal. Actually, the rebuttal is not, I think, pending peer reviewed. Is actually in preprint from MIT saying that no, I think there is a question of semantics. You're looking at the endogenous gamma in the brain, but you can interpret it as the 40 hertz you're delivering is syncing with the 40 hertz, the gamma in the brain that's not presented. So, okay, let them debate it out. But the point is, if you go back to oops, this is going forward. What I showed here, look, if you deliver 40 hertz with a photobiomodulation device, like, according to our parameters, you're going to get endogenous gamma in the brain, exactly what you want. Okay, let's do the work and let's publish, because I'm hoping to shake the institution a little bit. But then there was another study that came out of China that is an animal study that did they did 1070 nanometers. They actually compared 10 hertz and 40 hertz. According to them, you can go into this literature that 10 hertz is actually better. In animals, the behavior improved. In other words, their cognition and memory. The the markers in amyloid beta were reduced significantly more in the 10 hertz than the 40 hertz. So it's all up for debate. I think in our next clinical study, we will combine 10 hertz and 40 hertz. It's not to miss out on anything. Now positioning. is for years, actually, we're talking about since maybe around 2010 or so. Harvard, the group at Harvard actually published imaging on Alzheimer's disease brain and found that there were accumulation of lesions and beta amyloid in the default mode network areas. And these orange ones are really nodes of the default mode network. So that is actually the original reason why I position them on a default mode, because if you understand photobiomodulation, it's meant to help with the recovery of the cells of the neurons. Therefore, hopefully it will reduce the lesions, and it does a number of other things in those localized areas. Now, confusion sets in when you look at okay, a little bit. I want to go back to an image, something like this. You put in one point, the whole brain responds, right? So why do you bother to have on that particular point? Now, the reason is there is another study, I didn't have the time, imaging as well, by a Japanese group, showed that actually there's more, if you pay more attention, there's more activation around where the light source is on the brain. Say if you put it on the dorsal medial prefrontal, there's a lot more there than other places. Okay, then there's a reason for having them positioned on the default mode network. And it also tells us a couple of things. You don't need LEDs all over, particularly when there's not enough power density, but you can achieve both the localized area and whole brain response by having enough power and targeting in these particular areas. So the future, I've touched on this, is going to be personalization. We will be releasing this, but our future studies are going to use a lot more of the NeuroPro anyway, because we want to look at what happens if you try to, how about incorporating the cross-frequency coupling between theta and gamma, right? How about theta? I will be publishing this hopefully in one of my future papers. How about theta in the hippocampal area, which is, particularly indicated for memory, and gamma in the neocortex? How about that combination? Gamma, there's literature published showing that there is improvement in brains that have theta gamma coupling in the prefrontal areas. So let's look at that. Okay, this is a repeat. And I would say, if for the time being, you can understand more on the pathophysiology of photobiomodulation with this piece of literature that I did. Okay, I want to thank everybody. Thank you for listening, and I'm open to Q&A.
All right. Hey, Lou, we'll get everybody unmuted. I have a quick question for you.
Sure.
When you're looking at whether it's 40 hertz or 10 hertz, how long would you say the exposure should be? You were talking about 3 minutes, 4 minutes, 5 minutes. What's the ideal time for, say, doing 40 hertz and then doing 10 hertz if you're going to do both on the same day?
I would actually, this question came to me this morning too during our research meeting for development of protocol. I would allow the brain to respond to one of each, given enough time. So I would do, if you want to do two, I would do, say, one in the morning, one in the evening. I would
Gotcha.
start with 10 hertz because 10 hertz, okay, I haven't shown it in here, but in some of our other work doing the experiments looking at the effect of pulse frequency on living cells and on tubulin as they aggregate to become microtubules. Microtubules form the structure of the cells. 10 hertz seems to be destabilizing. I think this was actually published in our paper on the living cells. So it's kind of like priming your cells
Hmm.
to be open, you know. And then when you deliver 40 hertz, 40 hertz, believe it or not, also pulse frequency. I think it's a wrong notion to think that. I'll explain a little bit later. 40 hertz is stimulating more than 10 hertz. Actually, in our 2017 paper, when we looked at gamma into the brain, we didn't have enough uh, space or word count to say that actually we saw more inhibition, global inhibition with the 40 hertz. Um, so there is, which also can be interpreted as more control, which is a good thing. While we see an uptick in the power of alpha, beta, gamma, we also see inhibition. Now, here's the thing that I'm seeing. When we put these the LEDs on the nodes of the default mode network, we are supposed to stimulate the default mode network, right? Especially when you close the eyes, 10 hertz, you're also in the alpha state. Yes, you are with 10 hertz alpha, but we are noticing a switch at 40 hertz. Now, here's the interesting thing that we notice as we do these studies with meditators. At 40 hertz, the brain starts switching from the default mode to another network, the salience network, which can be associated with the brain of the meditators. They actually, when you are in the zone, whether you are in sports or whatever, or you're meditating, you're in another state, like a bliss state, you're actually not in the default mode network. You're actually out of it. And what's happening? I was really confused for some time. And then I did some experiments, you know, because I said, okay, understanding the literature of meditation, we really don't want to be in a default mode. So let's move this stuff, these modules out of the default mode into other places. And then we noticed that the meditators were actually switching the brain state at much higher frequency. We're talking about 120, 400, and so on. So when we did that, it was a total failure. It does not seem to work. So which means that it kind of reconciled with the hypothesis that after 40 years, you switch away from a default mode network. When you put on a default mode, you're actually doing it correctly. At higher frequency, you're switching away, you know, from the default mode to, say, the salience network, which involves I think, the insular and the anterior cingulate cortex. And so that's why it seems to be working. So we don't have to move this stuff around. So that's a long answer to a short question.
No, that's good. And would you say, like, should sessions with each of those be 5 minutes or 10 minutes? What are you shooting
Okay.
for an actual time frame?
Okay, I showed you that, let me go back here. Where is that? Okay. This was actually 5 minutes treatment.
Okay.
Now, 5 minutes actually is enough to get you there, and it seems to stay there even after you stop. In this case, 15 minutes after you stop, and this is consistent with, you know, the subject. We're still doing more. Now, why do we need 20 minutes? Now, what you see is a response in electrophysiology, so electrical signaling. You can probably see that same thing here in blood flow. But I feel that if you're looking for something more for the long term, you're looking at physiological changes. Now, when you talk about physiological changes, you're starting to talk about, you know, modification of proteins, modifications. You're doing this gene transcription process. You're modifying mRNA and so on. how long does that take when you start talking about modifying your physiology for a more longer term outcome? Now, there was when you go back to the discovery of the DNA, the double helix, and so on, you know, that's in the 60s. A couple of years later, there was a group in Caltech that wanted to see. I won't go into the, you know, how they did the experiment, but it wanted to see how long it took to modify the gene. It took 20 minutes exactly. Okay. Now, that was actually on E. coli bacteria, but on humans, it's going to take hours and so on. So it's got to be a repeated process. So we have to distinguish between what we see in electrical signaling or even blood flow and improvements in your physiology. That's why I think the 20 minutes is a good thing. Some people might say, oh, you know, you look at an analogy of cooking. I've actually said that before, you know, cooking turkey in the oven, you don't want to blast it and, you know, get it done. You only get it in 5 minutes, couple of minutes or 5 minutes, you only get burned in the outside. So it gets time to cook in. That's a metaphor that we
Hmm.
can cite as an example.
Great. Well, thank you. I'm going to see if other people may have questions, as we know we're getting close to the top of the hour. So thank you.
Yeah, I got a question. It was mentioned recently that sometimes with the Parkinson's patients, peak gamma can be slower, let's say around 34 hertz. And the question would be, would it make sense to start training, let's say at 35 and then shift up to 40, or just jump in at 40?
I don't see any problem in jumping at a 40. So jumping at 40. Actually, it's an interesting point because the peak gamma for even Alzheimer's disease, even as we age, it seems to be lower too. So if 40 hertz, you know, 40 hertz is a frequency that's been brought up and discussed very much in the literature. So I think it's a good benchmark. There was a study that's done in University of Alberta that we work with. They actually found the strongest signaling between cells is actually 39 hertz. So it is close, but you know, 40 hertz is a pretty good number, I think. Yeah.
Just a quick question. What was the wavelength for that wrist or that pain unit
Oh,
that you're
this
going to be
this
using?
is using 810 nanometers.
810. Okay.
Yeah. Mm,
All right. Perfect. Look forward to that.
thank you.
Thank you, Lew.
If this, okay, I don't know if there's another question. I just wanted to say, actually, I tried to get another slide in from, but I did on my laptop for some reason. My, you know, the Dropbox was not syncing. But we are doing a pilot study for MCI right now, mild cognitive impairment. And it's a pilot that's going to a large pivotal trial to FDA approval. Okay, I don't want to go into the whole story, but the data is being written up right now. I think hopefully it will be published without too much wait. But we have seen, and this is done with imaging using MRS, which is magnetic resonance spectroscopy. So you use the spectroscopy to identify biomarkers. So some of the things are coming out pretty interesting. The MMSE, you know, the main thing was on the biomarkers, but the MMSE reading for cognition actually improved. But some of the markers were quite telling. They actually. So what I'm saying is, we are not seeing the improvement just in in clinical outcomes, but we can actually see the changes in the biomarkers too. So in this biomarker, we look at this INS, the lactate, NAA, also trail making AB. Trail making also shows significant. Trail making is when you follow literally the trails and connect the stuff. Is a presentation of cognition that also was significant. So just want to let you know there's ongoing work with MCI and Alzheimer.
Okay. Well, Lew, thank you so much for taking the time to
Of course.
talk with us today. I'm sure people will contact you directly at Vielight with information about some of the products you have. And if you get any papers published you'd like to share with us, if you shoot those to myself or Richard, we're very glad to put them on our listserv if it's okay to do, and
Okay.
share the research that you're doing. I found the one you mentioned on TBI, so I put that out there, and we're glad to help you in any way we can to further the research. So thank you so much for taking time today to talk with us.
Okay, everybody. Thanks so much for attending.